Hello everyone. In this video, we are going to study about the structure of antibody, which is a topic of class 12. So welcome to the channel Learn with me, and I am Shilu Patel. So antibodies are the protein molecules which are generated in the living body, and this specifically inactivates antigen molecules. To understand it, let's take an example where there is a person and he has been attacked with the virus which is causing chickenpox. This virus can be anything. It can be any microorganism, it can be a bacteria, fungi, anything. I have taken an example of chickenpox virus. So whenever an antigen will attack in our body, the immune system will be stimulated and the T cells and B cells will also be activated. These activated B cell will start producing antibody which are specific for these antigens. Antigens are any foreign substance that is causing infection in our body, and these antibodies will try to kill these antigens. So this is a role of antibody, and we have already discussed in detail in our previous video. You can get the link in the description box. So in this video today we are going to study about the structure of antibodies. Now, chemically, antibodies are globular proteins. Globular means they have spherical structure. Just have a look in this image where you can see these protein molecules are spherical in shape, and that is the reason why the name is given globular proteins. Now, they form about 17 to 20% of the total proteins which is found in the blood serum. Blood serum is the liquid part of our blood. Now these antibodies belongs to the class of gamma globulin serum proteins. They are found in the serum, but the class is gamma globulin serum protein, and that is the reason why the name is given immunoglobulins. In short, we can represent it as IG. These immunoglobulins are produced by the lymphocytes. We already know there are two type of lymphocytes, B cells and T cells. So B cells are producing antibodies. Antibodies are made up of two long chains which are called heavy chains and two short chains which are called light chains. So we can represent them as H2 and L2. H here stands for heavy chain, 2 is the number of chains. Similarly, L for light chain and 2 is the total number of chains. So in total, there are 4 peptide chains are present in the antibodies. These orange colored part represents the constant region which is responsible for determining the class of immunoglobulin. This is very important. Constant region is responsible. And what are the different classes of immunoglobulins? It is like IgA, IgG, IgE, IgM and IgD. We have already discussed the different function of these different kind of immunoglobulins in our previous video. Now after that, this blue colored part is called variable regions, and this is the antigen binding site where the antigens are actually binds to this surface. Now these heavy and light chains of the antibodies are connected to each other with the help of disulfide bonds. There are total 16 disulfide bonds are present in between these antibodies. Now the question arises, what makes these antibodies so specific for their antigens? Actually, the variable region is responsible for making it specific. This variable region contains a unique sequence of amino acids that forms the antigen binding site which recognizes and binds to the specific antigens. Now the N-terminus domains of the heavy chains are called Fab region, that is fragment antigen binding region, and it binds to the specific antigens. Whereas the C-terminus domain of these heavy chains are called FCE region, that is fragment crystallizable region, and it interacts with the cell surfaces. Now what is these cell surface receptors? These are actually the proteins which are found on the surface of certain cells. As you can see in this image, FC receptors are the protein molecules, and it can be seen in certain cells such as B cell, macrophages, natural killer cells, neutrophil, basophil, eosinophil, etc. Okay, so these proteins are found in the receptor cell. So the C-terminus region of the antibody binds to the effector cell, whereas the N-terminus antibody binds to the antigens. Is that clear? Now the second role of this C-terminus region is to activate the immune system. So just like heavy chains, light chains are also having two domains. The first one is responsible for recognizing the antigen sequences, and the second domain is responsible for stabilizing the structure of antibody. In both these chains, they have antigen binding site, and this region is responsible for stabilizing the structure of antibodies. And now this is a handwritten notes of structure of antibodies. These are different type of antibodies. I'm quickly changing the slides. You can pause the video and you can take a screenshot. Now it's a quiz time. So the question is, what makes antibodies so specific for their antigens? Answer me in the comment section. Kindly like, share and subscribe the video. Thanks for watching Learn with me. Bye bye. Take care.